Okay, so the next one we'll look at is the plant hormones called gibberellins. And the idea about gibberellins is that they cause stem elongation. So super long stems. Now, they've been able to sort of see this in nature where things have gone slightly awry, and there's a fungus that's kind of taken hold of some rice plants and has been producing gibberellins in high quantities, and that actually caused rice plants to become extremely tall. And that gave them this idea, actually, gibberellins were to do with stem elongation. So what they then found is that when they were to apply, say, a small plant like a cabbage, so typically a cabbage is kind of a small plant, when they apply gibberellins to this, what you end up is kind of a very strange plant, in that it's very long stems with its cabbagey appearance as so. So you're getting stem elongation. So we know then that gibberellins do cause stem elongation. Now, they wanted to know what actually happens in nature, because actually it's all very well smearing paste on a plant. It might not be in the right quantities that you find naturally occurring within a plant. So they wanted to do a bit more work to do with that. So the next thing they did was to look at naturally occurring plants, so tall varieties and dwarf varieties, mutations, to understand what was going on. So what they looked at then was, say, a small plant variety, it may be something like a pea plant, and a tall plant. So small and tall. Now what they noticed that within the tall plants there was a particular type of gibberellin, GA1, gibberellic acid 1, that was in high levels. So the quantities of GA1 in this tall plant were high. But in the small plant, the quantities of GA1, gibberellic acid 1, was low. So that gave a bit of a clue that something must be going on. And when they started to analyse this, they realised that there was a mutation in the small plant that stopped a conversion of gibberellic acids along the pathways that occur. So there must be something going on a biochemical level that involves conversions of gibberellins to make this particular gibberellic acid. So when they started to look at pathways, what they noticed is that there's lots of chemicals involved in the production of this GA1 that they knew was kind of really key, and they found the pathways, and there's many steps involved, you can have a look at your book, but this pathway here, where you've got one chemical being converted into another chemical, so lots of different types of gibberellic acids, was kind of key to understanding the whole process. So this one was something like GA12 aldehyde, and some stages before here too. So what they knew with these tall and small plants is that this enzyme here that was involved in the production of GA1 from GA20 was not being manufactured correctly. So there must be a gene mutation problem that was causing these small plants because they weren't tall. And they actually found this gene to be called something like the L gene. And this gene manufactured an enzyme that would do this conversion job of GA20 to GA1. So that was kind of interesting. It kind of explained this idea of why these plants were small and why they were tall. They then looked at other plants that had also small characteristics and tall plants, varieties, and they looked at this idea that some of the plants had a problem with possibly this enzyme here. Actually it was this one here, wasn't it? Conversion of GA12 aldehyde to GA12. So another mutation in these plants here. So what they then thought was, well, okay, these plants here, these small plants, can do everything potentially, but they just can't do the last step. And these plants here can probably do everything, but they just can't do this step. So what if we grafted those plants together? What if we took a plant that couldn't do this first step here, this first, say, mutation, I'm going to call it mutation A, and we grafted to it a plant that could couldn't do this mutation over here, this mutation that's been labelled L gene, then really this pathway should work. So when they did this, the actual results they got was a nice stem elongated plant, a nice tall plant with a long stem. So they were able to prove then a couple of things. They were able to prove that gibberellins were involved in stem elongation to get tall plants, and they also could prove that it wasn't just one gibberellin, there's many types that are involved in a metabolic pathway that was needed for that final stem elongation to take place. So hopefully this will help understand some of the experiments that took place to do with stem elongation.